Bueno, a ver, gente, hola, buenos días, bueno, buenas tardes ya, de hecho es afternoon, porque ya hemos empezado después de las 12. Os voy a presentar a Iker, que como hay confianza entre nosotros me ha dado su currículum justo hace 5 minutos. Me lo sé bien su currículum, pero las fechas me bailan. Entonces, bueno, Iker hizo la tesis conmigo. Bueno, a ver, vamos para atrás. ¿Dónde va esto? Hace muchos años. Bueno, Iker, de hecho, aquí no lo pones, pero fuiste JAE Intro conmigo antes. Y en 2008 a 2012 hizo la tesis doctoral aquí, en el Museo de Ciencias Naturales, con anfibios, si no recuerdo mal. Puede ser, ¿no? Eso. Luego se fue con mi amigo Axel Meyer a la Universidad de Constance entre 2013 y 2016. Luego, esto para animar a la gente, que sepan que uno hace la tesis y empieza a viajar y a viajar y a viajar y a viajar. Después estuve en Alemania hasta el 2016, otros 2 años en Uppsala, en Suecia. Volvió aquí, 2 años como Juan de la Cierva, y es la I, de incorporación. Y luego se fue a Alemania otros 2 años más, a Göttingen, o sea, estamos ya en el 22. Luego se fue a Hamburgo, al Leibniz Institute for the Analysis of Biodiversity Change, que en realidad es un museo, el Museo de Hamburgo, donde estabas implementando lo molecular la museogenómica en el Museo de Hamburgo. Y después de todo ese periodo, por fin el Estado tuvo a bien en darle una Ramon y Cajal y ya estás aquí. O sea que esta es su charla de vuelta al museo como ya Ramon y Cajal. Y en unos cuantos años más espero que científico titular. Pero como te tomas tu tiempo para todas las cosas, pues tampoco va a ser inmediato, vamos. Pues nada, y entonces lo que ha hecho en todo este periplo último por ahí, dando vueltas por ahí, dejó los anfibios ya, y ha estado analizando transiciones evolutivas muy interesantes, no solo de animales, sino también ya veréis que ahí hay algas y origen de plantas, y también a nivel del origen de los eucariotas y todo esto. Y eso es lo que os va a contar hoy. Así que adelante, cuando quieras, Iker.
Gracias. ¿Se oye bien? ¿Qué? Ah. Ahora sí. Y nada, muchas gracias, decía, por la introducción y es un placer estar aquí con todos vosotros, presentaros la investigación que he estado haciendo estos últimos años. Me hace especial ilusión porque, como decía, esta ha sido la casa donde comencé con los primeros pasos en la ciencia y bueno, I should switch to English because the talk will be in English, and I wanted to apologize because my voice is not doing so well today, and hopefully we can manage to fill 1 hour. So my talk is about phylogenomics and evolutionary transitions. And as you know, during the history of evolution, there has been several tipping points that have been very crucial for biodiversity as we know it today, including, of course, the origin of prokaryotes, the origin of photosynthesis, then symbiosis has been also very important, for example, for the origin of eukaryotes. Several lineages have become multicellular, and there has been multiple transitions between different habitats, especially for continents, the terrestrialization process of plants and fungi and animals have been very, very important for the terrestrial environments as we know them today. So evolutionary transitions could be defined loosely as events in evolution where you have a change in organization, for example, origin of multicellularity or symbiosis. Could be also changes in the Bauplan with the very characteristic Cambrian explosion. But also changes in habitat can imply very important drastic modifications. So one of the main points I wanted to stress today is that evolutionary transitions are great models for understanding the processes that generate and maintain biodiversity. And this is because during these evolutionary transitions, we often have high rates of evolutionary innovation, and this often leads to evolutionary radiations. So we have high levels of natural selection and diversifications. But these very same processes that make them very interesting make them also challenging from a phylogenetic point of view. And so the fact that we have often are very ancient events, and these events happened, there has been rapid radiations. This leads to the typical example when phylogenies are difficult to resolve. And we often get these blurred trees that are difficult to resolve. But at the same time, we all know that phylogeny is key whenever we want to compare anything between different species. And so we need to spend a bit of time and effort in improving these phylogenies if we want to learn something about these evolutionary transitions. So I will talk about 3 different stories. The first one is the origin of photosynthetic eukaryotes. So photosynthetic eukaryotes, or commonly known as algae, are very, very diverse. Some of them have primary plastids, which originated by symbiosis with cyanobacteria, for example, red algae, green algae, and glaucophyte algae. But then we also have huge diversity of photosynthetic organisms that have higher level plastids. These are acquired through symbiosis with different eukaryotes. For example, kelp or diatoms are 2 of the most famous ones. Traditionally, we have assumed that the origin of plastids occur in the ancestor of these 3 lineages, red algae, green algae and land plants, and glaucophyte algae. And these are called the archaeplastida, or old plastids, and these are primary plastids. From a phylogenetic point of view, it's very clear when you do a phylogeny of the plastids here, together with a bunch of cyanobacteria, the monophily of these 2 lineages is very strongly supported. But with the arrival of phylogenomics, when people started to build phylogenies on nuclear genomes, so in this case, the nuclear genome is the host of the plastid, then they did not recover the monophily of archaeplastida, here shown in green, in many cases. So in this, what is very interesting here is that we can have monophily of plastids, but this does not require the monophily of the host of the nuclear genome. And there are several biological scenarios where this is actually possible. One of them is, for example, the secondary loss of the photosynthetic activity. Or we could have serial endosymbiosis. This is a process by which one of the lineages could get a primary plastid through symbiosis with cyanobacteria, and then the other lineages could get it through symbiosis with this first eukaryotic lineage. So the plastid could be transferred here. Or we could even have cases of multiple symbiosis, independent symbiosis in the 3 different lineages. And actually, if this we represent here the phylogeny of cyanobacteria, you can very quickly see that we can have 2 independent symbioses from closely related cyanobacteria, especially if these are not sampled or most probably extinct. We would not be able to identify that these are 2 different events. In the phylogeny, it will appear as a single event. So in order to explore this, we started by asking ourselves why different studies recover such different relationship among these three different Erkeplastida lineages here are shown in green. And we chose 4 datasets that have been generated by independent labs with different numbers of taxa and genes. But what you can see here is that we have very different topologies. In these 3 cases, Erkeplastida are not monophyletic. They also differ in the specific relationships, and the support is generally very low. In this 4 dataset, we have a monophily of the 3 Erkeplastida lineages, and the support is really high. So this is quite surprising. So what we saw is that the differences in taxon and gene sampling do not seem to explain why these different topologies are different, but what we can see very clearly is that the 4 dataset that has the strong monophily of Erkeplastida, you have some genes that have a very high support for the monophily of Erkeplastida. Here I'm showing the difference in log likelihood scores for each gene in the dataset for the monophily of Erkeplastida in green and against in red. And you can see that in most cases the differences, genes are not able to say whether they are for or against because the difference in log likelihood are rather small. In this 4 dataset, the signal is very, very strong. The difference is about 100 times stronger, sorry. And we have these genes. So when one goes into the alignments and looks at the gene trees, you can actually see very clearly that there is a mix of paralogs in these genes of nuclear genes, but also plastid and mitochondrial genes. And this is because the authors of that specific paper did not explore the data enough, and they inadvertently included these paralogs here. What happens is that when you remove these very clear paralogs from 6 genes, you actually lose the supposed monophily of Erkeplastida in these 4 datasets. And so we end up with 4 datasets that do not support the monophily of Erkeplastida, but also they recover different topologies, and they are lowly supported. So what can we do now? What we tried was to combine the data, being very careful about combining the things, removing all the paralogs and all the contaminations, and using rather complex models of evolution, we were able to recover the monophily of Erkeplastida here shown in green. But there are 2 surprises here, and they are these lineages that are shown in black, which you can see they are included in Erkeplastida. And these are Rhodelphidia and Pycozoa. So these are 2 lineages that have been described, they have been known only very recently. Rhodelphidia is a secondary non-photosynthetic eukaryote, so it does not produce, it does not generate photosynthesis, but it does have a plastid without a genome, which seems to be involved, for example, in a heme biosynthesis. So this is easily explained by secondary loss of the photosynthetic activity in Rhodelphidia. The case of Pycozoa is much more interesting because they do not have an evidence, they do not have a plastid, and we also find no evidence of ancient plastid. So if they lost the photosynthetic activity or the plastid, very often what we can see is the presence of genes of algal origins in the nuclear genome. This is something very common that you can see here in all these photosynthetic eukaryotes. This is called the endosymbiotic gene transfer. When the plastid is acquired, very often many of the plastid genes go into the into the nucleus, and this allows the cell to take control of the plastid. In the case of Pycozoa, here in green, you can see that they have very, very few genes that could be explained by endosymbiotic gene transfer. And actually, they are very similar to other species that have no plastid. So it's rather surprising. So summarizing, we said that the presence of plastid without the nucleus, without a genome in Rhodelphis is easily explained by secondary loss here. But the absence of plastids in Pycozoa is a bit more complicated to explain. And we would have 2 possible hypotheses. One of them was the traditional hypothesis of a single endosymbiotic origin of plastids in ancestor of all Archaeplastida and 2 secondary losses in these 2 lineages. But we could also imagine the scenario where we have 2 independent origins or actually serial endosymbiosis for the origin of plastids, one in the ancestor of green algae and glaucophyte algae, and another one in the ancestor of Rhodelphidia and red algae. And as you can see here, in this scenario, Phycozoa would not have any ancestral plastid. So take-home messages are that we, of course, need larger data sets, but also cleaner, better data sets to resolve these complicated relationships, phylogenetic relationships. So data quality is very important, especially for these relationships that seems to be difficult to recover. Looking at cell biology and genomic traits, we could say that perhaps the most parsimonious hypothesis remains the single origin of the plastid in the ancestor, followed by 2 secondary losses. But it's also true that, and we have here a clear example of whenever we discover new deep lineages, this can have a huge impact on how we understand the evolution, in this case, for example, for plastids. And both Phycozoa and Rhodelphidia have been only recently discovered. So I would not be surprised if we are able to find new lineages that change this picture further. And I have also to say that we could even reevaluate the cell biological features, the homology or suspected homology of the different structures in the cells, that maybe we could see evidences for the alternative hypothesis of either serial endosymbiosis or independent origin of plastids. This remains to be seen. And here, so this is the paper that I presented. It was the study that presented these first crucial steps towards understanding the evolution of early plastid evolution. And here I put another paper where we explore the secondary plastids in a bunch of independent eukaryotic lineages here. This is how, for example, kelps or diatoms originated, through secondary, tertiary, and even quaternary endosymbiotic events between different eukaryotic lineages. But I don't have time to talk about this. What we can talk about is, so once algae had the ability to photosynthesize, as we saw in the previous slide, there has been also a transfer of these complex secondary plastids among very different eukaryotic lineages. What we can also see is that other major transitions happening during the evolution of eukaryotes, for example, the origin of multicellularity, happen in many different lineages independently, and even habitat transitions. Here is a big phylogeny of eukaryotes, and you can see in orange are species that appear on soil, then we have freshwater, and then marine euphotic and marine aphotic regions. And you can see that there are multiple transitions between these different environments. And here we come to the second story, which is about the origin of plant multicellularity and terrestrialization. So the earliest land plants appeared around 600 million years ago, or the earliest terrestrial algae, actually. And maybe this looks like a quite unglamorous moment of evolution, because you have this kind of algae that live on rocks and so on. But actually, if you imagine, this is also quite epic moment, because the stressors that happen in the terrestrial environment are quite different from those that these algae are exposed to in the marine environment or a freshwater environment. So we have this terrestrialization process during evolution around 600 million years ago, until we arrive to the hypothetical last common ancestor of land plants around 500 million years ago. And here we have the typical divide between tracheophytes and bryophytes. So real land plants with 3D multicellularity, different tissues, and much more complex body size. But there is much more in this lineage of the streptophytes to explore. So the lineage that gave rise to land plants is called Streptophyta, and this belongs into the green algae. In Streptophyta, we have 6 lineages that are at paraphyletic rate with respect to land plants. And they are very interesting because they differ enormously in complexity. Many of them are unicellular. Some of them are, then they have different kinds of multicellularity. Some of them simple, like simple filaments, but we also have 2-dimensional flat structures like Collocytophyceae and 3D structures like Cara, for example. They also differ a lot in their environment. Some of them are aquatic. Many of them live actually in soil or on rocks, on tree barks, can form algal mats. And so there is also quite some diversity in the stressors that they have to respond to. As in the previous case, actually in all the examples that I will present in this talk, having a robust phylogeny is key in order to compare the features in these different species. And this already happened in 2019 in this very big effort, where they recover that the Cycnematophyte algae are the sister group to land plants. And this was quite a surprise because they are by far not the most complex algae. People for many years assumed that Carophyceae or Collocytophyceae could be the sister group to land plants simply because they are morphologically more complex. But actually, phylogenetics leave no doubt here. However, there is much more to unpack in the phylogeny of streptophytes, and here I'm showing 2 studies where we generated dense sampling for 2 of the classes of Streptophyte algae, Klebsiella and Cycnematophyceae. And not only we improved the systematics of these groups of algae that did not, they don't get enough attention from the taxonomic and systematic point of view. So I think it's nice that we try to improve also that, because actually there is a lot of interesting features that we could learn from here. Not only we improve the phylogeny, but then this dense sampling allow us to perform ancestral character reconstructions that are more reliable. And here we have the ancestral characteristic reconstruction of multicellularity in streptophytes. Here we have the clade of embryophytes, so the real land plants. And what you can see here is that many of the streptophyte algae have some kind of multicellularity. And actually we can trace back this multicellular ancestor even, so for sure, to the common ancestor of embryophytes and this lineage known as Klebsiella that are mostly filamentous, but probably also to the ancestor of Streptophyta. And another interesting case here is in this study, we increased the sampling of Cycnematophytes, a sister group of land plants. And you can see here that we not only recover multicellular ancestors for streptophytes, but probably the ancestor of the Cycnematophytes was unicellular. And we also see up to 7 independent origins of multicellularity within this clade of Cycnematophytes, which is quite crazy. So this suggests, first, that there has been a potential for simple multicellularity growth in streptophytes for at least 1,000 million years. And the fact that we see so many transitions between uni and multicellularity suggests also that it might not be so difficult to do that. And we think that this is due to the presence of some ancient machinery for multicellularity. We also have, there was a very nice paper of talking about signaling this ROP gene that shows that in Klebsiella mediophytes and land plants, these proteins have a specific motif that allows them to become multicellular. Whenever you remove this motif, then plants are not able to to be multicellular. And the frequent gain and loss of multicellularity also suggests that there might be independent genes in each of these independent events. We also try to recover the origin of terrestrialization or terrestrial species, and you can see here that embryophytes are clearly terrestrial. They are what we know as land plants, but we also see that there is an independent origin of terrestrialization, at least in the lineage of Klebsiormidiophytes. These Klebsiormidiophytes are mostly terrestrial filaments. And here I'm showing some comparative genomics that we try to study in order to understand the genomic basis of this water-to-land transition in plants. And it's a general approach of comparing genomes in order to identify this genomic basis. And again, I cannot stress enough the need of a robust phylogeny in order to do this. Here we sequence the genomes of 4 strains of Cycnema that were the first multicellular cycnematophytes to be sequenced. And long story short, what we find in the ancestor of cycnematophytes and land plants, we find an expanded presence of gene families related to phytohormone signaling, transcription factors, membrane transporters, and cell wall. And we also find protein domains and gene ontologies related to specialist metabolism, cell adhesion, cell wall, and transporters and immunity. So what this seems to suggest is that the ancestor of cycnematophytes and land plants was already quite complex and able to perform very complex signaling. Specialist metabolism is very important in plants because it's the main mechanism by which they react to the environment. They cannot run like animals, most animals. So they have to respond to the environment using phytohormones or specialist metabolism. Here I want to talk about gene co-expression networks. So as an introduction, in case someone did not know what the gene co-expression network is, basically this is based on transcriptomics, and the idea is that you try to identify the gene modules, so the different genes that are co-expressed, upregulated or downregulated in different conditions. And you express this as a network. And these genes, on the first hand, they are responsible for that process or for that response. For example, if we have a gene module that is upregulated under heat stress, then we know that these genes must be related to that one function. And it can also help us to actually identify biological processes for genes that have not been annotated. As you know, in many model organisms, many genes are not annotated functionally, so we don't know what they do. But the fact that they are co-expressed with other genes that we know are related, for example, with heat stress, then this allows us to somehow pinpoint to some functions. So using this, we generated a bunch of networks, and perhaps the most interesting pattern here was the co-expression of genes that are signal receptors, signal transducers, and target genes. Sorry. So what does this mean? This makes reference to what has been called the plant perceptron. The plant perceptron is an idea used from machine learning, actually, to explain what plants do in the environment. The idea is that you have different receptors here that obtain information, for example, about temperature, light conditions, nitrogen, phosphorus, drought conditions. And these receptors, they get information from the environment. Then this information is transduced into a complex network of protein-protein interactions that are used to modify this information and to integrate it. And so what this leads is to a final answer that is integrated the different receptors into one or more target genes. And so the fact that we find the co-expression of receptors, transducers, and targets in the same network suggests that in signematophytes we also find this perceptron. And this is what allows signematophytes to respond dynamically to terrestrial stresses. And so if we know that the sister group of land plants have this capability, this suggests that it was most probably present in the common ancestor. So in other words, that this dynamic process for responding to terrestrial stresses predates the origin of land plants. So summing up, we have that phylogenomics allow us to reveal a very old potential for simple multicellularity in streptophytes. We find also frequent transitions between uni and multicellularity, particularly in signematophytes, the sister group to land plants. And then I presented some comparative genomic analysis that suggests that there is a deeper origin of several traits that have been considered to be land plant specific, including complex signaling, phytohormones, or the dynamic response to terrestrial stresses. And here we come to the last story, which is the conquest of land by vertebrates. You know that land vertebrates are tetrapods and originated here. This includes amphibians, mammals, crocodiles, birds, turtles, lizards, and snakes. And there has been a long number of studies trying to recover one specific phylogenetic question, which is, what is the sister group to tetrapods? This is a classic example that has been studied from many different point of views, including paleontology, but also morphology and molecular systematics, including several pioneering works by Rafa Zardoya. And what we did here, so basically for the relationship between tetrapods, lungfishes, and coelacanth, there are three possible topologies that we have. And basically there have been so many studies that all three hypotheses have been supported in one or the other study. So what we try to do here is to bring this question into the era of phylogenomics. And what we were able to do is not only to support this one hypothesis as the most plausible one, so the sister group of tetrapods would be the lungfishes. This was not new because, as I said, many studies supported this hypothesis before, but also this one and this one. The only difference, or let's say our contribution here, is that we were able for the first time to statistically reject the two other hypotheses. And not only that, but we also show that these hypotheses were consistent with systematic error. So they are generated by systematic error. What is systematic error? Systematic error is one of the main tricky questions in phylogenomics, because when you have many genes, small model misspecifications can add up and generate systematic error. This is a typical example where you have two different topologies based on phylogenomic datasets that are both strongly supported. You have no way of deciding which one is the best one, right? And this happens because of systematic error. Systematic error, it's very important to differentiate between precision and accuracy. So phylogenomics gives us very high precision, which means we have a lot of information to resolve the phylogenetic questions. And in analogy with a statistical distribution, precision means that my distribution is very close to the mean. So we have a lot of power here. But this does not tell us anything of whether my mean is representative of the true population. And this is accuracy. And so therefore, phylogenomics needs to not only have high precision, but also high accuracy, and we need to test the presence of systematic errors. So in the case of languishes and coelacanths, this would be my topology 1, sister group of tetrapods and languishes, and this would be a very precise estimate for the wrong topology, in this case, topology number 3. So we extended this phylogenomic pipeline, and we were able not only to resolve with confidence the sister group of tetrapods, but also 2 other cases that have been contentious in the last years. And actually, again, this is nothing new, because many studies, including molecular phylogenetics by Rafael Sardoya, have already resolved the monophily of Lysamphibia, and also the sister group of turtles with Archosauria. But here we revisited with the phylogenomic methods, and I have to say that these 3 questions that have been, or we thought that they were resolved maybe 20 years ago, were put into question again with the first phylogenomic studies. So this was our motivation to bring these questions into the phylogenomics era, and we were able to show that yeah, life is beautiful, and the hypotheses that we knew from previous studies are stable. Just that in the beginnings of phylogenomics, maybe people were not careful enough about what they were doing. Once we resolve this phylogeny of vertebrates, we can start to do comparisons. And here I show a recent study where we look into the evolution of the karyotypes, of ancestral karyotypes in the tetrapod ancestor, but also in the previous vertebrates. And what we can see here is that, so we sequence the genomes of the 3 landfish lineages that exist today. And this is not a simple thing, because landfishes have huge genomes. So compared to 3.6 gigas of human genome, landfishes have between 40 and 70 gigas. So it's really a lot. They are really big. It's really difficult to work with these genomes. And one interesting result in this study is that actually, actually, even if the genomes are extremely large, these have been bloated with the transposable elements. Despite these gigantic genomes, the synteny is more or less conserved to the ancestor of tetrapods, but also to the ancestor of the chordates. So we have these conserved blocks of synteny in the ancestor of chordates that are maintained in the 3 landfish lineages with relatively few changes, which I find it very crazy, considering that they have more than 90% of the genome is full of transposable elements. When we reconstruct the evolution of genome size, we can also clearly see that there are 2 independent origins of gigantic genomes in vertebrates. One is landfishes, and one is salamanders. And when we look into the transposable element landscape, we can also see that these gigantic genomes originated due to independent transposition of independent proliferation of different transposable elements. Here is the landscape of landfishes, and here would be the axolotl, for example. We also find several genes that are related to that are positively selected for functions that are relevant in the landfish, but we also find several traits that are quite surprising. One is, for example, the expression of SHH gene in the developing lungs in the lungfish that is very similar to that we find in amphibians. But we also find an expansion of gene families related to airborne odorant receptors and expansion of vomeronasal receptors in lungfishes, at the same time that we have a contraction of gene families related to the reception of waterborne odorants. So these characteristics are more similar to what we find in tetrapods compared to the case or the situation in fishes. And perhaps one of the most important innovations in this water-to-land transition of tetrapods was the origin of hands, or limbs, hands, and fingers. And here I show an example of how you can use Evo data together again with, of course, the robust phylogeny in order to understand these processes. And here we studied the expression patterns of genes that are related to the development of hand in tetrapods. And what we find here is that there is a medial region in the fin of the lungfish that is similar to what the condition in tetrapods. And this suggests that the ancestor of lungfish and tetrapods, they already had some kind of a primitive hand. So in terms of development, of course, this is not a hand, it's a fin, but developmentally, the center of the fin is homologous to the palm of our hands. And what we also find is the expression of HOXD13 here that is expressed in the posterior part of the fin in the lungfish, and in tetrapods, this has a dynamic expression during development that goes from your pinky towards the thumb. And this is diffused slowly from the pinky to the thumb. This dynamic expression of HOXD13 is not present in lungfishes. So this suggests that the fingers, which develop thanks to this dynamic expression, are an innovation exclusive of tetrapods and not present in the common ancestor. So recapitulating, we have a common ancestor with a developmental hand and secondary origin of digits in tetrapods. And here some take-home messages. Again, the importance of phylogenomics that allow us to resolve the lungfishes as sister group to tetrapods. I show also several examples of comparative genomics that allow us to understand the evolution of conserved synteny in Sarcopterygians, and also the independent expansions of transposable elements in lungfishes and salamanders that lead to giant genomes, as well as the presence of adaptations that we could think are beneficial for tetrapods, but actually happen in lungfishes. Again, we are talking about features that are believed to be tetrapod specific, but we often find the case that these have a much deeper origin. And here, another example of identifying the origin of hands and fingers with using Evo data. And to wrap up, some take-home messages. I hope that I could convince you that evolutionary transitions are very interesting as a model for understanding evolutionary processes. And one common feature is that phylogeny is often difficult to resolve because they are deep and ancient branches that we need to resolve. And this requires to have not only more data, but also better data and more complex models that allow to model the heterogeneities that happen in molecular data. And generally, the use of comparative genomics is a very powerful approach to understand the genomic basis of these different transitions. And generally, comparative omics and Evo, which can provide very important insights into these major transitions. And with this, I would like to thank you for your attention. Of course, all my collaborators and funding agencies, and I would be happy to answer any questions. Thank you. ¿Qué? Ah, estaba aquí. No.
Okay, thanks, Iker. Time for questions. I will start one. Apart from bringing back to very melancholic days in my life, I have a question. which is, well, you have demonstrated here that many of the features or the genes and everything predates the transition so this idea, the old idea that is still in evolutionary biology takes about key innovations triggering radiations. How much is true or not at all?
Yeah, that's very interesting. I think that the more we look into the details and of the different transitions, I think we are realizing that biology is more complicated, and yeah, I think it's unlikely that in many cases there are these key innovations that start a new radiation, because actually if you think about it, it makes sense from an evolutionary point of view, because what evolution does is to reuse things that are there and use them in new forms. So I think it's a general pattern that we will see whenever we look into the different transitions. For example, there has been a recent paper, they were talking about this pharyngeal jaw in cyclists. They also show that it's not the thing that explains the...
Okay, so there are no single key innovations that explains always transition, radiations, and probably because another nice place that scientists have used key innovations is in the origin of humans. So there are no key innovations probably. So it's a long transition, and many things were there before. Anyway, this was just to start.
Thank you very much, Iker, for this very interesting talk. You didn't have to convince me that evolutionary transitions are cool, but you reinforced me. I was particularly impressed by the archaeplastida, all these papers on the endosymbiosis stuff, and probably a stupid question, but you use the plastid genome as well for the phylogeny of, or can it tell you anything about the origin or multiple origins of these plastids?
Yes, thanks, Anna. Actually, no, we don't use the, what is this thing? We didn't use the plastids because we didn't use the plastids because plastids have a very clear signal for monophily. So this cannot be contested. It's very strong. The challenge here was to resolve this phylogeny using nuclear genomes. Exactly because of this, you can have the monophily of plastids, which you have, but you can have different scenarios for the evolution of nuclear genome, and that's the tricky part. No one ever doubted that plastid genomes are monophyletic.
Yeah, but can tell you anything about the potentially double origin of these endosymbiosis? If they fall into different categories, I don't know, I have no idea. Sorry. Probably not.
Maybe, I think no. I think not that much, I don't know.
But maybe when you have these gene networks working together, if this is a convergent origin, then you will have very different results than if it's a single origin, no? Maybe.
The multiple origin is not... there are no clear evidences for that.
I was not thinking in this. I was thinking in terrestrialization and multicellularity. But anyway, more questions?
Thank you for the talk. Very diverse and entertaining. My question regards the three hypotheses for tetrapods relationships, and is when you calibrate these phylogenies in time, in a temporal dimension, would that impact or benefit one of the three scenarios above the others, or it doesn't have that much of an impact?
So in general, phylogenetic topologies do not have a huge impact on the molecular clock analysis. And anyway, the confidence intervals that you get from molecular clock analysis are way too broad in order to distinguish between different hypotheses. So, yeah, I don't think that this is what you are asking, right? Yeah. I don't think that it helps.
And actually, I should add, since I know about this, that this was 360 million years ago, and he was testing the living sister group of the tetrapods, not the origin of the tetrapods, because the origin is a distinct lineage of Sarcopterygians, which is very clear in the fossil record and perfectly dated. More questions? Okay, well, thank you, Iker.
Thank you.